Okay, hi everyone. Welcome to this week's seminar. It's our last seminar of the semester, so yay. Professor Adams is out sick, so that's why I'm introducing. You usually never hear from me. I'm here to have our lovely introducer, Julie Meyer, to introduce our presenter for today.
All right, well, thank you everybody for coming and tuning in online. I'm very pleased to introduce you to Allie Colvin, PhD candidate in my lab. Allie came here. After this degree, she will be a triple gator. She did her bachelor's of science here in animal science and animal biology. She did a master's in wildlife ecology and conservation. Did a little stint at the USGS studying wildlife disease. And the work she did before her PhD actually has resulted in 4 publications, some as first author and some as co-authors. And she entered the interdisciplinary ecology program under a graduate school preeminence award. And I wanted to share just a couple pictures with you to give you an idea of just how well-rounded Allie is as a scientist. And she's been an absolute pleasure to have in the lab. So whether it's doing field work underwater, sitting in the dirt processing samples, doing lab work, and presenting at conferences. Allie's always professional and a good example for our lab. And these things, in addition to these things, I have to say that one of her most impressive skills is her commitment to developing her computational skills as a PhD student. So what I'm showing you here are Allie's wall hours. So the amount of time that she has spent on Hypergator. And for those of you who don't know, it's our on-campus supercomputer, which is also the fastest supercomputer owned by a university in the US. And Allie has put in so many hours using the Hypergator that has equivalent to almost 8.5 years of computational hours. And in addition to this, she has pursued 5 UF-based trainings in bioinformatics and computation. Some of those were a series of classes. She attended a Joint Genome Institute workshop out in Walnut Creek, California for a week. And she also was awarded a competitive position in a competitive week-long NSF-funded bioinformatics and science communication workshop. She's also learned to become a science diver during her PhD and has contributed to other projects besides what she'll tell you about today that will result in at least 3 publications. So with that, I will turn it over to Allie.
Wow, Julie, thank you for that wonderful introduction. Hi, everyone. I'm Allie Colvin, and I'm excited to speak with you this afternoon about some of my dissertation research examining the role of microbes in Orbicella faveolata disease dynamics. This one? There we go. Okay. So Orbicella faveolata is our mountainous star coral. It's found throughout Florida and the Caribbean, and it's important as a reef-building coral species. However, unfortunately, it's affected by numerous diseases. So this includes yellow band disease, black band disease, white plague, Caribbean ciliate infection, bleaching, and stony coral tissue loss disease, which is what I'm going to really be talking about today. Stony coral tissue loss disease emerged off Port Miami in 2014 and then spread north and south along Florida's coral reef, where it's now considered endemic. And it has since spread throughout the Caribbean, where it impacts over 30 of our stony coral species. It's really been unprecedented in terms of its geographic spread, the number of species impacted, and its virulence. It's characterized by acute to subacute tissue sloughing. So you can see how quickly some of these lesions progress, leaving behind the bare white skeleton. So in less than a month, this coral has actually lost over half of its tissue due to this disease. Affected reefs can lose up to 60% of their live coral cover, and this outbreak has led to the functional extinction of some highly susceptible species. And that's led some to conclude that SCTLD is likely the biggest mortality event to impact the Caribbean. We do see that Orbicella faveolata is intermediately susceptible to SCTLD, but really there's a wide variation in how this disease presents in this coral. So some may be susceptible OFAVs, they'll develop lesions. These may progress rapidly and require a lot of intervention, versus our resistant corals can actually exist on that same reef as infected conspecifics for years without showing any signs of SCTLD lesions. We don't know the pathogen of SCTLD, but what we do know is that microbes are implicated in SCTLD pathogenesis. So, for example, we consistently see the same group of microbes again and again and again across multiple disease studies. And we found that the application of antibiotics is effective at halting lesion progression. So here you can see a coral that did not receive antibiotic treatment. It lost all of its live tissue and unfortunately died, versus this coral in these two panes on the bottom here actually received antibiotic treatment and SCTLD lesions halted. This has led to widespread applications of amoxicillin on Florida's coral reef and throughout other affected areas of the Caribbean as an intervention strategy. So here you can actually see me applying some antibiotic paste. It's a little tricky, but I'm holding a syringe and it kind of comes out in this paste that you apply along the disease margin. However, this is a very time and labor-intensive process, and we don't really know the implications of these long-term applications of amoxicillin to our corals. Therefore, it's really critical that we investigate mechanisms of resilience in corals to find alternative intervention strategies. And with that, I'd like to introduce you guys to the coral microbiome. So coral actually exists as a holobiont or a meta-organism that includes the coral host, the algal symbionts, and microbes, which are found in this thin layer of mucus along the surface of the coral throughout their tissue, all the way down into the skeleton. And the coral microbiome performs important functions for the holobiont, such as the cycling of nutrients, the synthesis of amino acids and vitamins, occupying niches or producing antimicrobial compounds that prevent exogenous microbes from pathogens from being able to invade, and the production of antioxidants, which may actually confer resilience to bleaching. So because of this, I don't think it's a surprise to say the coral microbiome does play an important role in modulating coral health and disease. Corals really exist in this state of homeostasis with their microbial community. So here on our y-axis, we have relative abundance, and we would expect to see in a healthy coral microbiome a high proportion of metabolic mutualists performing all of those important functions that I just talked about. However, in disease, the microbiome shifts to a dysbiotic state known as the pathobiome. And this is really characterized by higher abundances of opportunists, which are microbes that take advantage of disease tissue, copiotrophs. This rise of opportunists may be associated with altered holobiont function, not performing all of those important roles that we had talked about before, and therefore promoting disease development. Interestingly, it may not be an invasive pathogen, which could be transient, but rather this pathogen may trigger a shift in the microbial community, causing interactions that push what are normally considered mutualist or commensal microbes towards overgrowth or production of virulence factors. So what this means is that disease may not be just the result of the presence of a pathogen, but virulence may occur at the functional gene level rather than the microbial species level. So the disease by pathobiome paradigm essentially involves multiple microbes acting together to result in the disease phenotype, and it really comes down to the functions of that community. So we would expect that genes associated with pathobiome establishment to drive corals towards disease susceptibility. For example, prokaryotic toxin production. Toxins are substances that are harmful to cells and can result in host tissue damage. Toxins have previously been associated with coral tissue loss and are implicated in SCTLD. Quorum sensing is a form of microbe-microbe communication. Quorum sensing pathways may regulate virulence factors like toxin production, and quorum sensing has been implicated in coral disease previously and may regulate the progression of white band disease in acropora corals. Additionally, biofilms, biofilm formation. Biofilms are bacteria that are embedded in an external matrix attached to a surface. You can kind of think of it as almost like a slime layer. Biofilms have been associated with coral diseases such as black band disease. The cyanobacterial mat is essentially a biofilm that kind of smothers the underlying coral tissue and causes lysis. And biofilms are likely a reservoir for SCTLD. So we would expect some of these functions to push a coral more towards disease susceptibility. Additionally, microbial functions can also promote resistance to disease. For example, the production of antibiotics, which are compounds that can kill or inhibit the growth or reproduction of bacteria. As I've said, antibiotics are effective at halting SCTLD lesion progression, so we would expect these to promote disease resistance. So our overall question became, how does the coral microbiome impact SCTLD presentation in Orbicella faveolata? And we would expect our resistant corals to have a symbiotic microbiome dominated by metabolic mutualists and have more genes, abundant genes involved in antibiotic production. Versus our susceptible corals, we would expect to see a dysbiotic microbiome dominated by more pathogens and opportunists, and more genes involved in toxin production, quorum sensing, and biofilm formation. So kind of really tackled studying these OFAV-associated microbes 3 different ways. First, I quantified a single pathogen, Vibrio coriolyticus, in OFAV corals. Then I took a different approach by evaluating microbial community gene functions in OFAVs of different susceptibility and resistance. And finally, I took what I learned from these first 2 chapters to study a different disease that emerged during the same time as SCTLD and characterize the microbial communities of that disease. So that leads into my first chapter, quantifying the presence of coral pathogen Vibrio coriolyticus in corals of varying resistance to SCTLD. Vibrio coriolyticus is a known coral pathogen. It's the etiological agent of several marine invertebrate diseases, including bacterial bleaching in coral and acute Montipora white syndrome, as depicted here. Their genomes possess numerous virulence factors, so that includes toxin production genes, quorum sensing pathways. They even possess the full gene cassette to produce hydrogen cyanide. But what I'm going to be focusing on today is the Vibrio lysin-like zinc metalloprotease, VCPA toxin. And what this actually does is it degrades photosystem 2 of the algal symbionts and causes tissue lysis in corals. It's also been found that co-infection with Vibrio coriolyticus, VCOR, exacerbates SCTLD. So corals that actually tested positive for this toxin had higher mortality and faster lesion progression rates. So you can see in this top row here, that coral tested positive for the VCPA toxin, lost most of its tissue, not doing so good. Versus the 2 rows on the bottom tested negative for the VCPA toxin and had much slower SCTLD progression. So the question really became, what is the relationship between the abundance of Vibrio coriolyticus and resistance to SCTLD? When we think about our gradient of resistant to susceptible corals, we would expect our resistant corals to have lower abundances of the VCPA toxin gene and our susceptible corals to have higher abundances of the VCPA toxin gene. So we sampled 90 OFAV colonies from across Florida. So in Southeast Florida, the ecosystem conservation area there, and also in the Lower Keys. And it's important to note that these cores were taken from healthy tissue on the colony. We sampled them 3 times in May to June 2021, August to September 2021, and again in February to March 2022. These colonies were also fate tracked for 5 years and kind of divided up into these dynamic disease states. So they were part of these monthly monitoring efforts, and at each point they made a note about whether the coral had disease or not and treated it if they did. And we divided them into these dynamic disease states or resistance categories. So unaffected corals up here never showed signs of SCTLD throughout the entire 5 years, and we can think of those as our high resistance corals. Corals with active SCTLD disease had lesions both before and after sample collection. We can think of those as our low resistance corals. Will be affected corals never had SCTLD prior to sample collection, but showed lesions after. And recovered corals had SCTLD before sample collection, but those lesions actually resolved prior to when the samples were collected. And we can kind of think of those as some intermediate levels of resistance. We extracted DNA from these cores and quantified the VCPA toxin gene using droplet digital PCR. So what did we find? Here we have our VCPA gene abundances on our y-axis and our dynamic disease state on our x-axis. And overall, during that first sampling period, abundances of Vibrio coriolyticus were pretty low, and there were no significant differences across our resistance categories. This pattern held true for our second sampling period, as well as the third sampling period. We did see higher abundances during this time frame, but they were not significantly different across disease categories. The third sampling period had significantly higher abundances of Vibrio coriolyticus, and this was true for both the coral ECA and the lower keys, indicating that this is a region-wide phenomenon. We also wanted to investigate prevalence of Vibrio coriolyticus, so that's the proportion of corals that tested positive for the VCPA toxin gene. And during the first sampling period, we saw 68% of our corals test positive, but during the second time frame, we saw that number rise to almost all of our corals at 95%, and that was maintained going into our third sampling period. So our first sampling period actually did have significantly lower prevalence compared to sampling periods 2 and 3. But the question really is, how does that correlate with our resistance categories, or those dynamic disease states? So here, each horizontal line is a coral, and it's colored by its dynamic disease state. And you can see how the corals changed from Vibrio coriolyticus negative to positive over time. So these is the group here that changed. And as you can see, all of our disease states were represented. So there doesn't seem to be much of a relationship between V-core infection status and our resistance categories. And again, during the third sampling period, not too much changed there. So overall, dynamic disease state does not seem to be correlated with V-core infection status. So what's going on at each of these time points, right? That seems to be kind of the difference here. The first time period is characterized by high rainfall and moderate inlet flow, but that second sampling period is when we started seeing the highest temperatures. So some of the genes that Vibrio coriolyticus requires to infect a coral are temperature regulated, and so that may be why we're suddenly seeing this increase during that second sampling time frame. The third sampling period had the lowest temperatures and winds. This is also the dry season when we expect salinity to be higher, and some strains may be more responsive to salinity for growth compared to temperature, and so that could be why we're seeing higher abundances during this time frame. So getting back to our question, was the relationship between the abundance of V-core and resistance to SCTLD? Not much. We found pretty low abundances in both our resistant and our susceptible corals. However, it seemed that seasonal environmental conditions seemed more connected with these V-core abundances than our susceptibility to SCTLD. However, I think it's important to note that disease progression is likely the result of more than a single microbe or virulence factor, and therefore I decided to look at all of them. And that leads into my second chapter, evaluating microbial gene functions in OFAV resistance to SCTLD. The overarching question of this chapter really was, how are microbial functions different between corals of varying resistance? And going back to that hypothetical framework I outlined before, we expect our resistant corals to have more abundant genes involved in antibiotic production, and our susceptible corals have more abundant genes involved in toxin production, quorum sensing, and biofilm formation. For this chapter, I actually only focused on the samples in the ECA, that Southeast Florida Ecosystem Conservation Area outlined here. Lower key samples are being analyzed by our wonderful postdoc, Cynthia Becker. It was a lot of data, so we ended up just kind of divvying up between the two of us. These were kind of divvied up into 3 locations within the ECA, the north, middle, and south. And these coral cores were also typed for algal symbiont composition by qPCR, and that was done by folks at the University of Miami. We also only focused on the first sampling period, and rather than looking at those dynamic disease states, we only analyzed corals that had SCTLD or not prior to sample collection, so previous SCTLD versus no previous SCTLD. So those coral cores were used for shotgun metagenomic sequencing. Metagenomics is sequencing all of the DNA in a sample. So that would pick up our coral DNA, our algal symbionts, and the microbes. And really the question we're getting at here, what this allows us to do is look at which microbes are there and what they can do. So we're really getting down to that genetic functional potential. So I scraped coral tissue with sterile forceps and scalpels, extracted DNA with the QIAGEN DNA microbiome kit, and this preferentially extracts microbial DNA, and sequenced those libraries on an Illumina NovaSeq 6000. And what we got back was 5.3 billion sequencing reads, hence the 8.5 years that I spent on the supercomputer. This was a lot of data to work with all at once. So I executed a bioinformatic pipeline to enrich for some of those microbial genes in silico. And I'm not going to walk through all the steps here, but what I really want to emphasize is out of those 5.3 billion sequences, our final data set was just shy of 35,000 genes. And those genes had functional annotations, taxonomic assignment, and predicted counts greater than 2 across all samples. So what did we find? Here we have a principal component analysis. Each axis represents an amount of variation, and each point represents a coral microbiome. So the closer 2 points are, the more similar their microbiomes are functionally. The further apart they are, the more different their microbiomes are functionally. The points are colored by algal symbiont composition, and their shapes represent the different locations within the ECA, and the borders around each point are whether the coral had SCTLD prior to sampling or not. And what we actually found is that the microbiome functions of these corals are different based on whether the coral previously had disease, its symbiont composition, and its location within the ECA. Though I do want to point out that the amount of variation explained by the symbiont composition in the location was much greater than what we saw for disease history, which explained about 5%. So I ran what I call, it's a corncob test to determine which genes were differentially abundant between corals with different disease histories. And I actually controlled for the effects of symbiont composition and colony location, since they're such significant sources of variation. So here we have our 85 differentially abundant genes. On our x-axis is our effect size, and our y-axis is the broad functional categories that those genes belong to. Anything to the right of the dashed line was found to be more abundant in corals with previous SCTLD. To the left of the dashed line is more abundant in corals with no previous SCTLD. And unfortunately, most of these genes could not be assigned a functional category, and that's kind of a bummer to draw ecological conclusions from, but I'm going to focus on 2 of the categories that did have more genes with functional assignments. So first, nucleic acid replication, recombination, and repair. Our resistant corals, these abundant genes were primarily involved in mobile genetic elements in DNA stability, which could both serve as a source of rapid genetic adaptation and maintenance of genetic material through time, respectively. And our differentially abundant genes in our susceptible corals, we actually saw a lot of those involved in replication and growth under changing or stressful conditions, which is kind of what we'd expect from opportunists. We also found a lot of genes to be different that were involved in carbohydrate transport and metabolism. So in our corals with no previous SCTLD, these genes were really involved in energy production and starch utilization, so symbiome maintenance, versus in corals with previous SCTLD, we found a lot of those genes to be involved in degrading structural carbohydrates, such as those in the algal symbionts, and host immune evasion. Again, kind of what we'd expect to see from opportunists being able to utilize different carbon sources and persist in the host through time. We also found 4 quorum-sensing genes to be differentially abundant, and they were all more abundant in corals with previous SCTLD, but we did not find any differences in toxin production, biofilm formation, or antibiotic production genes. Given the proposed relationship between SCTLD and toxin production, I don't want to spend a ton of time on this, but we did identify 12 toxin production genes in our metagenomes, and each one of those dots is essentially a coral metagenome that we, if they're colored in, a coral metagenome that we identified that toxin production in. So the genetic potential for toxin production and host damage is always there, even if they're not different between corals with different disease histories. So getting back to our overall question, how does the coral microbiome impact SCTLD presentation in Orbicella faveolata? We didn't see differences in the toxin genes, the biofilm formation genes, or the antibiotic production genes, so that's a little bit different than what I expected. What we did find is that genes were involved in carbon cycling and metabolism rather than virulence factors like toxins and biofilm formation. These may play a bigger role in coral response to SCTLD. Mobile genetic elements may provide genetic variation and greater adaptability potential in our resistant corals. Increased potential for bacterial growth under changing or stressful conditions kind of implies that microbial blooms or overgrowth may play a role in pathobiome establishment, and the potential for quorum sensing is enriched in our SCTLD susceptible corals, implying microbe-microbe interactions are also important in pathobiome establishment. So with all of that, this leads into my 3rd chapter, investigating microbes in this potentially novel coral disease. And, just these poor corals, they can't catch a break. While SCTLD was going on, we started to see other OFAVs in the Keys developing these lesions that looked similar but behaved somewhat differently, which led us to believe that this could be a potentially novel disease, which has been named Orbicella acute tissue loss disease. So we think it's probably different than SCTLD because it progresses more rapidly than typical lesions found in OFAVs. And it only appears on Orbicella faveolata versus SCTLD, which has kind of a broad host range, and it has a much lower antibiotic efficacy rate. So whereas amoxicillin treatments are really effective at halting SCTLD, they only work about 1/3 of the time in ODOLD, which is this new disease. Very fun to say. So that kind of got us questioning, is this truly a new disease? Is it a more virulent form of SCTLD? So our question really became, well, are the microbes different? If we know that we find the same microbes consistently in these different SCTLD disease studies, is the microbiome of ODOLD distinct from SCTLD? So I kind of had 3 goals, which was to compare the microbial diversity patterns between healthy and ODOLD affected corals, to determine differentially abundant microbes between healthy and ODOLD disease lesions, and to contrast those with what we've previously seen in SCTLD. So OFAV colonies were sampled from 3 sites in the Florida Keys. They were collected from ODOLD disease lesions, and that's going to be labeled as DD. We also had samples that were taken from healthy tissue on those same disease colonies. So those are paired samples, and those are given the label HD. And we collected samples from healthy, unaffected corals located at those same sites, which are given the label HH. It's also important to note that these were all actually collected from an SCTLD endemic zone. These samples were collected in January 2024. So SCTLD had been in the area 5 to 7 years, depending on the site. So it had been there for quite a while. So we collected coral mucus in sterile syringes, extracted DNA with the QIAGEN D&EZ Power Biofilm Kit, and amplified the V4 hypervariable region of the 16S rRNA gene for microbiome sequencing. That's a really good gene for being able to taxonomically identify which microbes are present, and sequenced all of that on an Illumina MiSeq. And just for comparison, that was 1.6 million sequences. So this was a much more manageable data set to work with. And that resulted in 1,202 amplicon sequence variants, which we can think of as unique taxonomic identifiers. So those are unique microbes within sequences within that data set. And what did we find? Here we have a PCA representing about 35% of variation in the microbial community structure. Again, each point is a coral microbiome. The closer those points are to each other, the more similar their microbiomes are compositionally. The further apart they are, the more distinct their microbiome compositions. Each point is colored by health condition, and the shapes represent the different sites. We found that microbiome composition was significantly different between health conditions, but particularly our disease lesion samples compared to our HH, our healthy tissue from healthy corals. Though site was considered significant when we did post-hoc pairwise comparisons, we didn't find any sites to be different from each other, so we didn't differentiate by site for the rest of these analyses. Here we have a box plot showing the distance to centroid for each health condition. So the distance between each point and the center of its group from the previous plot. And what we see is that the disease coral microbiomes are more dispersed or less similar to each other than the healthy coral microbiomes. And this can be an indicator of dysbiosis in these lesion samples. I also ran a CLAM test, which is a multinomial species classification method test. Essentially, it classifies species as specialists of different habitats or generalists. So I'm just trying to quickly orient you to our next figure, which is the output of that test. On each axis, we have microbes abundance in healthy or diseased tissue. So in our upper left corner, we'll have our healthy coral specialists. In our lower right corner, we'll have our disease specialists. And along this one-to-one line is where we'll actually see generalists. About half of our ASVs were actually considered to be specialists for healthy coral tissue. And so we can think of those as our symbiome microbes. And additionally, about 15% of our ASVs were considered to be specialists for disease lesions. So we can think of those as our potential pathogens. I just also want to highlight that about a quarter of our ASVs are considered generalists, so they can be abundant in healthy or diseased corals. And these are commensals that may be behaving quite differently or functionally different between our healthy and our diseased tissues. I ran a DE-seq2 test to determine which microbes were more abundant in our disease samples compared to our healthy. So on our x-axis, we have the log fold change, and on our y-axis, we have the taxonomic assignment of all of those differentially abundant microbes, and they were all found to be enriched in our disease lesions. And I actually ran a comparison to look for sequence similarity to microbes that had previously been seen in SCTLD or belong to the same groups that have previously been associated with SCTLD. And these are all the ones that were either identical or really similar. So a lot of similarities. However, we did find some ASVs, these 5, that appear to be more distinct. They don't really look like what we've previously seen in SCTLD. I ran a nucleotide blast sequence search to NCBI GenBank to essentially look for how similar these microbes were compared to other things we see in the database to learn a little bit more about them. So on our x-axis here, we have health condition. Y-axis, we have the relative abundance of each of these 5 microbes in the microbial communities. And this Cribber habitans, I'm just going to kind of go through and tell you a little bit about the sequence matches we got. Matched really closely to what we've previously seen in black band disease and white plague. The Pheromonas was a good match for a bunch of sequences that came out of white plague in OFAVs. The Arantivirga matched some sequences from sediment and seawater, just kind of seems to be an environmental microbe, likely an opportunist. This mycoplasma was really interesting, though. It had only one match in the entire database, and that was from a white plague lesion in OFAV. This is a really interesting group of microbes because they are known pathogens in humans and other animals. They don't have cell walls, so antibiotics are actually less effective against them. And we really don't know much about what their role is in coral microbiome. So we think that this microbe in particular is worthy of future investigation in OFAV disease. And this Mycovibrionaceae matched some sequences from a deep sea octocoral. But worth noting, this is a group of bacterial predators, and their role in coral disease hasn't really been investigated either. So another exciting avenue for future research. So getting back to our main question, is the microbiome of otolde distinct from SCTLD? We did find that the microbial community was different between healthy and otolde affected corals, and there were indicators of dysbiosis in our otolde disease corals. They had an increase in some microbes, and some of those microbes were identical to what we've seen in SCTLD, though some, like the mycoplasma, were more distinct. So what did we learn? After all of this stuff, what did we learn? V-core abundance is not associated with SCTLD susceptibility, but is influenced by seasonal environmental conditions. The coral microbiome functional potential is different between corals with and without previous SCTLD, and some of those differences were involved in carbon utilization and quorum sensing. In otolde lesions display dysbiosis. While some of those microbes are similar between it and SCTLD, there are others such as mycoplasma that are distinct. So I hope at this point I've convinced you guys that microbes are associated with coral health and resilience. We've identified a few mechanisms that may promote resistance in these corals. However, it's important to note virulence potential does exist in the coral microbiome. We didn't find an association between bacterial toxins and SCTLD susceptibility, but the potential is there. And microbe-microbe interactions do seem to be important for pathobiome establishment. All of this to say, commensal microbes may become more abundant or switch to pathogenic roles under conducive conditions, such as the invasion of a primary pathogen or switching carbon sources. And overall, this work supports the disease by pathobiome paradigm, essentially that disease phenotypes in corals are the result of multiple microbes acting together and what those functions are. The broader impacts of this work, SCTLD and now otolde remain conservation issues for Florida and Caribbean reefs. Some of the resistance mechanisms we identified could be useful when we're looking at probiotic or good bacterial strains to combat these diseases. It can be used to develop assays for biomarkers of susceptibility and resistance in our corals, which will enable managers to prioritize resistant corals in restoration efforts, or maybe identify susceptible corals that require more intervention. And essentially, this work could be used to develop tools that will mitigate the impacts of future coral disease outbreaks. With that, a lot of acknowledgements, especially my advisor, Dr. Julie Meyer, and my co-advisor, Dr. Andrew Altieri, and my committee members, Blake Ushijima and Don Berenger. Big shout out to Dr. Cynthia Becker for all of her training in bioinformatics and professional development, all of the folks in the Meyer and Altieri labs, the members of the Resistance Research Consortium and otolde research groups in Florida Department of Environmental Protection and SNRE for their financial support to get this research done. PhDs don't happen in a vacuum, so there's just so many people to thank, all of the amazing scientists in the Meyer and Altieri labs. I'm a better scientist because of you guys. You inspire me with all of the great work that you do. The friends who bring a lot of joy when you have to step away from your science for a few minutes. The family who provide never-ending support, especially during the dissertation writing phase, which is fun. Ooh. And of course, who can forget Dewey the decimal dog, who's the best writing buddy. And with that, I'll take any of your questions. Yes.
A bit of questions to the connection. It was a nice talk.
Thank you.
So at in the last chapter, comparing the, I guess you call it little, but like the other
Yeah.
disease, you compare that to CTLD and suggesting that it has some similarities, but there's some unique kind of aspects of the microbiome. And that kind of implies that the CFP is sort of monolithic, and that there's sort of like kind of a microbiome that you associate with SFP that apparently is very consistent. But is it in fact? I mean, do you see variation like when you're comparing polls, different poll packs or different
Yeah, there's definitely variation in coral taxa geographically, but a lot of SCTLD microbiome studies do seem to still find some of these broad microbial groups present, and a meta-analysis actually pulled out a lot of those patterns as well. So even though there's a lot of variation just between individual corals within or across species, like we do see the same microbial groups pop out across studies. And so when I was comparing that, really just anything that had previously been associated with SCDLD. So those were sequences that came from the Meyer lab, some of our previous work, bioindicators, and the meta-analysis as well. So even if they weren't exact sequence matches, just looking for taxonomically similar to compare to kind of account for some of that variation. Does that answer your question?
Yeah, so I was just trying to get out whether the difference that you saw between disease and the CTLD is like more different than you see like within a CTLD.
Yeah, I think it is a little tricky because this was, kind of the first look at ODOLD, so to speak. But I think it's also really difficult to untangle because SCTLD had been there for so long, and some of those corals actually had SCTLD previously. So I think that could also add some of that noise to the data set. Yeah. Yes.
Thank you so much for a wonderful presentation and the science. It's really interesting.
Oh, thank you.
I was thinking of when we look at the different like diseases that's impacting you relate these with external factors. If we look at obviously there's climate change and then increase of temperatures, but we also relate it to the external factors like pollution, that really in a way impacts the corals. And if you work with the environmental conservation, is that what we can limit impacting that coral?
Sure. Yeah. Some of that wasn't really within the scope of my work. There's other folks that are more interested in kind of some of those environmental parameters and tying that to SCTLD. But we've seen more polluted areas have a tendency to have more expression of virulence factors in these microbes. I think plastic pollution has also been tied to kind of altered and potentially more pathogenic microbiomes in these corals. So definitely there are those ties there, and thinking about not letting those things get into the water and contributing to these disease, I think is really important. Yeah.
Because I think that your information gives good feedback back to the biologists on what should we do, what should we put in place to make sure that we're interested.
Yeah, absolutely. Anyone in Zoom land? No? Yes.
I'm curious, when you're in a poultry research group, if it's been seen anywhere else besides these specifically hotspots, and is the thought basically that it came, you hinted at it potentially could have evolved from that. I'm just curious if there's anything. I know your research didn't really talk in that, but just
in the grand scheme. Yeah. So it's a little bit tricky. What we actually ended up seeing, and I think I kind of highlighted. Let's see if I can even get back that far now. The computer is getting a little Sorry, I'm just trying to. There were actually some similarities to white plague, but we didn't have great microbiome studies at that time to really be able to characterize white plague. And so that's why maybe it looks a little bit different, but we don't have a lot of baselines to compare to. And that's really where some of the importance of this work comes in is kind of developing these strategies for examining coral disease so we can compare for the future and have better ideas of what are different syndromes versus not. Yes.
I'm also just curious a little bit about the background of odals. Was it identified? I mean, you said it basically looks the same as Skill D in the water, right?
I think what ended up happening is our collaborators were going out and doing regular SCTLD interventions, and they noticed lesions that seemed to behave differently than what they typically saw in SCTLD. So that's why we were like, is it a different disease? Is it not? And at this point, there's probably, we at least felt we had enough to say, like, give it a name at least, and further investigations into how different these diseases may be will be forthcoming.
So it's kind of based off like people monitoring the lesions. It's like
Mhm.
No more questions? Well, thank you, Allie. That was a great talk, and I learned a lot about coral that I didn't know before. So
Oh,
thank
great.
you.
That's the goal. Great. Thanks, everyone.